Hey everybody. What I want to talk about today is stuff to talk about like the middle part of the digestive system where we have the completion of the mechanical breakdown of the food and the beginning of the real hardcore enzymatic digestion and the start of the absorption. So the main deal today is going to be talking largely about like the stomach, the liver, pancreas, and how they're all regulated one another. But while one another is all regulated, largely due to the action of what the brain, as well as local hormones, as well as what hormones are causing to have happen. Okay, so there are these different phases that are recognized when we're looking at digestion overall, and we can kind of use the stomach as sort of a focal organ for thinking about this, meaning that so the three phases that are mentioned here are the cephalic phase, like up in the brain, the gastric phase, the stomach is the active part, and then the intestinal phase that comes later. And because we're talking about the stomach here, and particularly how gastric juice is released, this is this kind of summarizes a whole mess of events that are either going to cause gastric juice to get stimulated to be released, or are going to inhibit its secretion. So some things that are going to turn it on, like the sight of, or thought of food, smelling food, those things both are going to be sending reflexes down through the vagus nerve. They're going to start to get the stomach moving, start to get the stomach starting to roll a little bit, as well as starting to release the food. Because basically, the idea with these different phases of digestion is that the body is trying to prepare the gastrointestinal tract for wherever the food is going to be next. So in the just thinking about it phase here at the beginning, that's when that's going to start to rev up the action of the stomach. So it's not like the stomach's like going to get a whole bunch of food put in it like cold. So anyway, the gastric phase, as the stomach fills, that's going to cause both local reflexes as well as feedback on the medulla. That's going to have it start getting excited, start to roll more as it's getting more stretch put on it, as it's kind of filling up, and then also the chemicals that are in the food, as well as stuff that's being secreted by the stomach itself, and the decreasing the rising pH. That's going to cause the activity of the stomach overall to be kind of starting to really rock and roll. Now, in the end, what's going to start to dial back what the stomach does is a little bit of what happens when it starts to aliquot the food out into the duodenum. So as it starts to kind of fill that out, then the stomach's going to kind of dial back a little. So looking on like the inhibitory side, once you're full, then there's the satiation center that's located in the hypothalamus. As you start to be full, you start being like less interested in food. Or if you've just had a big meal, even if you smell somebody else's food, that's not going to get the stomach rolling because you're already filled up. So that will kind of dial back what the stomach's doing. Similarly, as the excessive acidity in the stomach, that's going to kind of slow down the acidity of the secretion of gastrin, which is a hormone. And also emotional upset, those things both together are going to kind of decrease its production. And again, as I was saying before, the stomach also starts to get dialed back, particularly with distension of food that goes into the duodenum. Some of that stuff happens like locally, but some of that's also fed back on the on the brain as well. So the take-home message from this whole figure here, even though it's complicated, if you kind of work through it, what you'll find is, the factors on the left, they're going to tend to increase the activity of the stomach. But then on the other side, these are the things that are going to be slowing it down, basically after a meal's already been eaten. But as the stomach's job is kind of ending, as the food's kind of worked its way from the stomach into the duodenum, that's going to make it, that's going to kind of decrease the activity that the stomach has. Now, if we look at the stomach, we've seen this in lab already, so I'm not going to talk a whole lot about its histology as far as the muscle walls go. We know there's that additional kind of diffuse oblique layer of the muscularis externa. But what I want to focus in on is particularly what's happening in the mucosa. That's the other part that's kind of modified a lot. So if we zoom on the mucosa, we see there are these deep invaginations that go into the wall that are called the gastric pits and the gastric glands. Honestly, I think we tend to refer to all of them as the pits, which is the ones that you can see at the surface of the mucosa. But where most of the actual active cells are happening is lower down, and there are three types of cells that are notable. First are the so-called mucous neck cells. Those are the ones that release mucus. Parietal cells, their job is to release the acid that goes into the hydrochloric acid that goes up for the chemical breakdown of food. And the other cells are called chief cells. Chief cells are cells that make the inactive enzyme called pepsinogen. Pepsinogen, when it gets broken up by hydrochloric acid, turns into the active hormone pepsin. And once you've made pepsin, that can also cleave also cleave pepsinogen, so it turns into the active form. So just like the fibrinogen that we saw when we were looking at blood clotting, here pepsinogen is the inactive form that needs to get activated either by acid or by pepsin enzymes. The other kind of cell that's here down in the gastric glands are what are called enteroendocrine cells, ones that are going to make the hormone called gastrin. That's going to be responsible for kind of sending the word not just to the stomach, but also to kind of rev up the activity of the pancreas and gallbladder. If we were to zoom in all the way on a single parietal cell, what we're seeing here is these cells that are basically almost have a similar set of reactions that we see when we were looking at the of hemoglobin or inside a red cell from before. Notice how the carbon dioxide that's in the interstitial fluid gets drawn in and then is turned into carbonic acid by the enzyme carbonic anhydrase, just like what we saw that's in the red blood cell. That carbonic anhydrase, so when carbonic acid dissociates, it's going to cause the hydrogen ions to get pumped out. And it does this in sort of this revolving door where it pumps out hydrogen ions but pulls in potassium ions from the stomach's lumen. And that way, it's going to kind of have an even swap electrically. But you notice also that potassium kind of wanders its way back out again just through a pore as well, through just a passive diffusion channel. That bicarbonate that's left over gets put into the blood from there, and also in another little swap with the antiporter that's going on between the bicarbonate portion and also the chlorine atom getting pulled in, so that HCl is both making it's constructed by basically stuff because the parietal cell is doing swaps between what's in the blood and the interstitial fluid in order to make hydrochloric acid. So up at the top, carbon dioxide is brought in, you make carbonic anhydrase, then when it dissociates, those hydrogen ions go into the lumen. On the other side, when the cell pushes out the bicarbonate ion, it's also drawing in chlorine, and the chlorine is transported through the cell and then also goes to the stomach lumen, and then they combine in order to make the hydrochloric acid. Now, the chemistry here is a little bit simplified, but notice here that basically the blood that's leaving the stomach, the blood that's going to that blood capillary is going to be more basic than what you would see in other parts of the body overall, because it's selectively dumping the bicarbonate into the blood. We'll see where that gets kind of neutralized on average later, but this is all part of that economy of carbon dioxide that circulates through the body. Now, this isn't super deep conceptually, but just to say that the waves that happen in the stomach, what we see is a series of both propulsive moves in order to kind of move the food from up at the top or the main section of the stomach down toward the pylorus, but also sort of grinding moves that happen where as you get closer to the pylorus, that's where the most vigorous mixing is going to be happening. And then finally, the so-called retropulsion, when the pyloric sphincter opens up, it does a couple things. Some of the fluid, some of that chyme that's now all liquidy, is going to get squirted into the duodenum, and the rest is going to get squeezed back into the stomach. And you'll continue this sort of squeeze, squirt, fill it back again kind of move until the stomach's basically empty. Now, this slide is talking about kind of slowing down the stomach after food has left it. So it kind of starts up at the top with the presence of fatty, hypertonic, acidic chyme in the duodenum. The idea is this is food that's been starting to get broken up, and it's also really acidic because it's just been mixed up with all that stomach acid. There are enteroendocrine cells inside the duodenum that are going to release hormones like secretin, cholecystokinin, vasoactive intestinal peptide, which are going to feed back on the stomach to kind of slow it down. Similarly, chemoreceptors, that's like the hormone way. On the other hand, chemoreceptors and stretch receptors, as the duodenum starts to fill, that's going to be connected with the rest of the CNS as well as gut neurons in order to kind of slow down the activity of the stomach. In this case, in particular, we're like dialing up the sympathetic division and dialing back the parasympathetic division going to the stomach. So the take-home message here, if you kind of work your way through it, is as the duodenum fills, hormones will feed back on the stomach to slow it down. Also, there are nerve reflexes that are also caused by stretch. This is a figure you've seen before when we were looking at the liver and the duodenum and the pancreas in laboratory. Just to remember that these accessory organs of digestion are ones that assist in digestion but aren't like a part of the alimentary canal. So what they're doing is they're just introducing different kinds of fluids to it. Of course, the liver is putting in bile, and the pancreas is going to be putting in the pancreatic juice. Whenever we often think about the pancreas as being active in digestion because it makes the hormones insulin and glucagon and somatostatin, but in addition to that, it also, but that's like what we call the endocrine part of the pancreas, and we've kind of talked that to death, I think. The exocrine part of the pancreas, on the other hand, here, these are substances that are getting secreted to a body surface through a duct. So that's what we call the exocrine part, and that's mostly going to be pancreatic juice, which is a combination of enzymes, like inactive forms of enzymes, as well as a bicarbonate-rich, like really acidic, basic fluid. So then when it meets up with the acidic chyme that's in the duodenum, it's going to neutralize it. So if people have been like vomiting, I talked about vomiting in the last video, people have been like vomiting from the stomach, or if you have acid reflux, that's acid that's going to be kind of irritating the esophagus. But food that's already gotten all the way to the duodenum, it's usually pretty well neutralized by that point because of the bicarbonate, but on the other hand, it has enzymes in it. So, it's irritating, but in a different way. So if we just kind of zoom in on the Pancreatic Cassini, we talked about these before when we were looking for the other stuff, when we were looking for islet cells. So the Pancreatic Cassini, that's like the exocrine part of the pancreas, and it operates the way that exocrine glands do, whereas there are these little granules that you can actually see in the slides called zymogen granules, and what those do is those are basically what's pumping out the precursors of enzymes as well as the bicarbonate. Just kind of switch gears to look at the liver. Liver is actually really interesting. It has this kind of hexagonal shape to it in its microstructure. And it's kind of weird that I don't know why I didn't look at this. We didn't look at this together in lab, but it's actually this kind of funny, these hexagons come from what happens in biological structures sometimes. Imagine if you were holding like a bunch of drinking straws in your hand. They all kind of pack together where like there's one in the center and there are like 6 other ones surrounding it. That's a structure that's really common in nature that we call hexagonal closest packing. That's what we see here. And the reason why it has like this kind of honeycomb structure to it is instead of being like true circles, all those little septa that divide up the different lobules are there to, they kind of end up squished into hexagons rather than into just really kind of leaving as circles. But if we actually look at a cartoon of what each of these lobules look like, what we see is a couple of different a bunch of different pipes we have to be concerned with. I'm going to zoom in a little bit on the structure here that's called the portal triad. Now what we have here is so you see like there's a bile duct, a portal venule, as well as portal arteriole all next to each other. Now, and those are occurring together at each of the like vertices of this hexagon. So if we zoom back out again, the portal vein down at the bottom, that is like food that has been absorbed from the digestive system that's got to, or from other places, that's going to be, that's coming from the digestive system because it's a portal vein. The food that's coming from the digestive system that's going to kind of go through customs here in the liver. So the portal vein is sending blood up as far as the slide looks. And if you look particularly on at the middle part here where it's kind of where the bile canaliculi are labeled, the blood that enters through those veins, it's going to kind of get squeezed in between these little folia in the these little sinusoidal capillaries that are there head toward the larger central vein in the center. So the blood works its way from each of those vertices toward the center of the lobule. And when it goes through there, that's where, because these are sinusoidal capillaries, this is where the interactions are happening with the blood. So this is where dead red cells are getting pulled out of circulation, where they're getting kind of ripped up. And you can see a little kind of a pull up here, these little bile canaliculi. That's just to show that those canaliculi make like a network that kind of goes up and down on the slide here. But anyway, the main thing I want you to dig is that the bile is being sucked from closer to the middle of the of these lobules out toward the periphery. I'm just going to change my view here just a little bit. So bile, even though we see blood leaving the bile canaliculi, leaving the portal venules, and then kind of working its way toward the middle, that's where the blood's headed. However, the bile, that's actually going the other direction. So those little bile canaliculi, like the little bile ducts, like starts here and then goes to this primary bile duct, and then the bile goes down. The blood is going in the other direction, blood's coming down and then working that way. So basically what we see is blood that's going through, that's going to be screened, is screened by these hepatic cells in these sinusoids, and that's the blood that's working its way from the perimeter up into the central vein. Or we see these little bile canaliculi, they're going to allow like the absorption of all that new bile is being made. So like the bilirubin and stuff that's formed in here, that's going to end up eventually reaching to like the right and left hepatic ducts, etc., to go down through the biliary tract. So the big thing I want you to understand here is this is like kind of as I promised at the beginning of the semester when we first saw the hypothalamic hypophyseal portal system. I promised that the portal systems kind of evolved, like kind of for use in the digestive system. They're kind of a consequence of the evolution of the hepatic portal system. So these portal veins, that's like food-rich blood coming in, but you don't just want to dump it straight into the general circulatory flow. You need to kind of send it through customs first. And that's what's happening here in the liver, is that the food, the new food that just got absorbed, it's going to get screened first. And also the portal arterioles are just kind of bringing regular oxygen-rich blood down to the liver. That's going to also be where like red cells are getting worked with as well. So like all of those like detoxification functions that happen in the liver happen through the flow of these two circulatory routes going through each of these little lobules. Okay, so enough of that kind of little diversion. I'm not going to talk about the morphology of the biliary tract or anything. I feel like we talked to that to death already when we were in lab. So just remember that even though the liver is always making bile, you're not always eating, so it's not always releasing the bile. So in cases where the bile is being made, it'll cause like your greater duodenal papilla will be, major duodenal papilla will be like sphinctered off. to prevent the bile from dripping off. So then basically, and it'll kind of back up and work its way up into the gallbladder. And we know that the bile that comes out of the gallbladder is like more concentrated, so it tends to be like greener compared to bile that's just been formed, which tends to be more yellow. Okay, about regions of the small intestine. These regions were recognized both by what they look like in other mammals, as well as histologic differences that we can see under the microscope. But for our purposes, when doing gross anatomy on a human, it can be a little bit tricky to distinguish them just in gross section. So oftentimes, figuring out whether, what part of the digestive tract you're looking at, you actually have to use a tape measure to figure out kind of where you are, because like your gut is actually longer in a dead person than it is in a living person because of the muscle tone. Anyway, as the food comes out from the pylorus and goes into the duodenum, that like first 12 inches, duodenum, so 12, 2 plus 10, the first 12 inches of the small intestine that hooks around the head of the pancreas, we call that the duodenum. There's some disagreement about this. I know there's some people out there will call it the duodenum. I grew up calling it the duodenum, and I think that's correct. But you do you. The jejunum is the next part of the small intestine, basically between the end of the duodenum up to the beginning of the ileum. The beginning of the ileum is like the trickiest part to find, I think, for me, but don't sweat it. But one thing you know positively is where the end of the ileum is. And the end of the ileum is here where the ileum meets up with the cecum of the large intestine in that little ileocecal valve. So one thing you can be glad for is that you can vomit from your stomach, you can vomit from your small intestine, but there is a point at which everything else goes out the other end. So if any chyme that's made it from the ileum into the duodenum, that valve there is going to prevent backflow. And again, that's a valve, not a sphincter, so it's going to be like a one-way valve, just like the valves we talked about in the heart. Anyways, that's like regionalization. But let's zoom in on what the microstructure is of the duodenum, or just the small intestine in general. We know from looking at it in lab, or just from being around, that there are a number of different efforts that have happened in order to greatly increase the surface area of the small intestine. I've heard estimates that it's like the size of a basketball court to allow the amount of optimize the amount of surface area for absorption. So first of all, rather than like a perfectly smooth mucosal layer, the lumen is kind of organized into these big circular folds that are formed up by the villi. Now, those villi, if you were to zoom in on the individual columnar cells that make up a villus, they also have the microvilli on their surface. And again, that microvilli, or what's also called the brush border, is basically there to increase the amount of absorptive surface area. Looking within like the guts of the villus itself, so basically all that kind of lighter staining stuff in the middle, that's all submucosa. So you can see both a loop of capillaries that swings through it, as well as a structure that's called a lacteal. Now, a lacteal gets a kind of green color in the cartoon here because it's like a beginning tributary of a lymphatic vessel. So we usually think about lymphatic vessels being something that prevent like edema, that kind of out of control swelling in tissues. But we also use it here because it's really good for absorbing fats. Oh, I suppose one other thing, there are both villi that stick up, so posts, as well as pits, like basically those little intestinal crypts where there are enteroendocrine cells. And enteroendocrine cells are sort of like those gastric cells as well, where they're going to be secreting hormones. Okay, now this sort of coming at you picture is supposed to represent what happens to the proteases, like the enzymes that are going to be intended for breaking down proteins. So there are a few main ones. When we're up in the stomach, we talked about pepsin, because pepsin is the hormone that's secreted by the gastric cells. There are three main types of proteases that come from the pancreas. One is called trypsinogen, and then chymotrypsinogen, and then procarboxypeptidase. So carboxypeptidase is those are like the three inactive forms of these enzymes. Now, just like how Pepsinogen got cleaved by HCl or pepsin to get turned into pepsin. A similar thing happens here, where these inactive forms work their way through the pancreatic ducts and then get released into the lumen of the small intestine. Doing that, these guys will bump up to to enzymes that are already on the surfaces of the microvilli. So microvilli are there for an increased absorption, but if you look at little purple proteases that are shown here, there are some that live on their surfaces. So what happens is, as the trypsinogen is released into the lumen, then it's going to bump up against the wall, and there are some of these peptidases that are kind of stuck to the surfaces, and that's going to turn the inactive trypsinogen into trypsin, or the inactive chymotrypsinogen into chymotrypsin, etc. And similarly, you see trypsin also being possible partners for activating the other ones. Trypsin is like the main enzyme that's released by the pancreas, or at least the main active enzyme here. But so because it can also cleave the other inactive forms of these enzymes into their active forms. Just to kind of zoom in here a little bit on what's happening with the secretion, with the digestion, enzymatic digestion and absorption. So the little protein fragments here, those are shown as like a couple little segments, like little beads of amino acids, just super diagrammatically. Those proteases will bind with those little segments of proteins and break them up into individual amino acids. So what I said in the last lecture about how your body is kind of fussy with the food it brings in, it doesn't just take the proteins as they are, like out of cow meat or out of pig meat or chicken meat or something. What it does is it breaks down those proteins, it digests those proteins into their simplest monomers and then builds up new proteins from them. Okay, so basically what gets absorbed are really small molecules now, particularly like amino acids in this case. So here, the amino acids are absorbed by active transport, so with ATP, then getting pumped in through transcytosis, and then they also leave those luminal cells in order to, they can kind of leave just by passive transport, facilitated diffusion, because they're in like high concentration inside the cell, so it's easy for them to leave. So, and then they get absorbed from the capillary from there. Fats work similarly, like with the same logic, but different players. So the function of bile acts like a detergent. If you remember detergent, like the surfactant that's in the lung, that helps to like break apart surface tension. Particularly here, it's going to be breaking apart fats. So we say that bile emulsifies fats by breaking up into little tiny droplets that are called micelles. Now, these micelles, when they get really small, are able to get absorbed into the luminal cells and then in turn absorbed into a lacteal. So basically, this is showing what it looks like on the side for looking if we're thinking about the absorption of fats. Fats are first broken up, by the bile salts, and then they're absorbed through the luminal cells by the mucosal cells in order to get put into the circulatory flow. Now, this figure is like a little complicated. I'm just going to kind of read through the steps here. But just to say that first, chyme that goes into the duodenum, we said is going to cause the release of certain hormones like cholecystokinin and secretin that's coming out of those enteroendocrine cells in the duodenum. Now, cholecystokinin it's a big word, but really the chole part refers to the liver, the cysto part refers to the gallbladder, and kinin is like increasing their activity. So cholecystokinin and secretin both are going to be revving up either the function of the liver or secretin to turn on the activity of the they both kind of turn in the activity of the pancreas. So so you both have, in the case of the pancreas, cholecystokinin and secretin is going to cause the production of the enzymes and the bicarbonate, and cholecystokinin is also going to cause the increase like the contraction of the gallbladder to let the bile out, and also the relaxation of the hepatopancreatic sphincter. So then the bile kind of enters that way. As the chyme leaves the duodenum, like as the stomach enters, as the stomach empties, puts the chyme into the duodenum, and then as the chyme moves past the duodenum, at that point all these processes shut off. So as the one of the things that happens is as the stretch on the wall of the duodenum decreases, so does the secretion of cholecystokinin and secretin. And similarly, during the you also have the vagus nerve, which will also kind of cause things to slow down a little bit as well. The last one was a little bit of a mess, but that's what I got. Throw that in for your feedback if you have any. Thanks.